Welcome back, everyone. For this afternoon, we have again two sessions. So both will be on modeling. The first one on plant uptake, so more related also to contamination, fertility, and then thereafter also modeling, but then more groundwater and soil systems. So yeah, as we have two sessions, Gilles, I will try keep track on the questions in the chat as well, but also we will try to keep focus on the time. Having said that, I leave the floor to you, and then I propose that we can start now. Okay.
Okay, thank you. Good afternoon, everyone. So, in today's lecture, I will begin with a small introduction. Then I will give some concepts which are linked to the plant uptake, mostly of contaminants. I will say some words about models of plant uptake, and I will illustrate with a case study that we are working on for the moment in my unit. So it is, of course, important worldwide. It's a worldwide issue to be sure that when you are producing food or feed for the animals, that they are safe of any risks. And there exists some books or some regulations in order to prevent and reduce the contaminations, such as the Codex Alimentarius that was built by the FAO. And at national levels or sometimes regional levels, there are also some institutions that work on the safety of what is in the plate of everyone's. So regarding the Belgian, it's the AFSCA in French. I don't remember the name in English, but it's the national agency which deals with the issues of contaminations. And they listed some kind of contaminants that can be found, that should not be found, but can be found in the food, such as dioxins and PCBs, flame slowers, paraffins, perchlorate, PFAS, heavy metals. But there are also nitrate, PAH, pesticides, and so on that are submitted to regulation. And the basic principles regarding the legislation is that the food containing contaminant should not be placed on the market. So mainly the regulation is dealing with the permission or not to sell products, not to eat, but to sell the products. And so the foods which are not compliant with the regulation should not be placed on the market. You should have a principle of precaution and try to keep the contaminants as low as can reasonably be achieved. And for some contaminant, the maximum levels should be set up in legislations in order to protect the public health. That's why you have a regulation, now EU regulation, that fix the maximum levels of some different kinds of contaminants that you can find in the food, such as mycotoxins, plant toxins, metals, and so on. So there are a lot of regulation. And if you look at the data surveys about contaminations, it's rather easy now to find data about soil contaminations. So there was recently a map of contaminations of cadmium at the European scale that was produced by the Europe. And so you see that there are places with less cadmium and the places with more cadmium and so on. But if you want to find the equivalent for food contaminations, it barely exists. I think it doesn't exist. because food contamination is usually more a problem of local contaminations and not a really large-scale issue. I put photos of illustration from a paper in the common journal of information in Belgium, and it was a case of local contamination of a farmer field by the local industry, and he had soil contaminations, and then he produced forage that he gave to his cattle, and the cattle was contaminated by, I think it was organic pollutant, and he had to kill all the animals. So these are real issues, but usually more local situations. So if we have a look at the main concept, so we will deal with the plant uptake. So in this image from the FAO, dealing with the soil pollution and the role of the plant can be diverse. The plant can help to make the phytoremediation. But here we will really focus on the uptake of contaminants by the plant and not on the issues of phytoremediation. First concept maybe remind the movement in solutions. So mostly the contaminants, when they are in the soil, they are moving in the solutions. And the main kinds of movements are first convection movement. So it means that the water is going from one place to another. And the contaminant, which is in solution or in particle, follows the flow. So it's called convection. And the other kind of flux is called diffusion. It's one element is going from a place where it is more concentrated to a place where it is less concentrated. So we have convection flux and diffusion flux. The convection flux, it's linked to the water movement, and so it is influenced by the plant, which is transpirating the water, and the transpiration makes a suction of the water from the leaf of the plants and to the roots, and so it is sucking water from the soils. And so you have a convection fluxes to the plant, in which the pollutant can go at proximity of the roots. And regarding the plants, there are two main pathways of entry for the pollutants. The one is through the roots, and so there will be a root absorption of water and going to the aerial part. And the other entrance is through the leaves, and so we will see these two pathways in more detail. So two main pathway of entry. If we look at the roots, the roots present architecture with different families of cells. The outer cells are constituting the epidermis. Then you have the cortex, then the endodermis and the pericycle, and then the xylem, which are the tubes by which the water and the solution can go from the roots to the upper part of the plant. So if we look at the fluxes of the water, it can go and follow what is called the apoplastic path. So there is a porosity inside the cells, and the porosity is constituting a network. All the pores are connected, and the water can go through these pores and go between the cells, not through the cells, but between the cells, and they can follow this path to go to the xylem. And following these fluxes, you will find the possibility for some contaminants, which usually are low molecular weight solutes. So it can be inorganic or organic, but small particles. They are able to follow this path and go to the xylem. It's kind of passive transport, so there's no activity. It's just following the convection flux, or also you can have diffusion from the most concentrated to the less concentrated content. Also, things are a little bit more complicated because in the endodermis you have a barrier which is called the Casparian strip or Casparian belt, and the water and has to go through this membrane. It's a bit difficult for the pollutant to go really through that one. And also the membrane cells through which the pores are connected have also a cationic exchange capacity. So it means that some charge elements which are in this porosity can be retained by the membrane, even if they are outside the cell. They can be retained by these membranes. So this is one fluxus, the apoplastic path, and another one is symplastic. So it means that the water is entering through the membrane of the cells, and it is migrating through all the cells. And so it means that the water and the contaminant must cross the barriers to follow this path. And it has usually a cost, energetic cost for the pollutant to cross these membranes. So it's much more difficult for the pollutant to go to the xylem. Also, there are in the cells, you can find usually vacuoles that are important organs within the cells in order to detoxify the contaminant. It can act as a reservoir of some of the pollutants, and some species that are tolerant to the high concentration of heavy metals are tolerant because they can store the contaminants in these vacuoles, and they are not having harmful effect on the other part of the cells. So if we look now at the membrane, so you have the cell which is here. The inside of the cell is down, and outside of the cell is up apart. And you can have here represented three different kind of transport. And first, the membrane is constituted of structure which have what is called a head, hydrophilic head. So these are phospholipids. So there is a head and a Q. And this part is hydrophilic, and that part is hydrophobic. So it means that it will be important regarding the behavior of organic pollutant or charged pollutants. So there are three ways for the elements to cross these membranes. So simple diffusion, I have a gradient of concentration. So here you have a lot of these points, and here there is less. So simple diffusion, the contaminants can enter slowly through the membrane. Here you also have a gradient of concentrations, but for some elements, there are transporter proteins that allow a more easy transportation through the membrane. So these are diffusions. So it's only the difference of concentration that drives the crossing of the barrier. And there is another one which goes against this gradient of diffusion. And it means that the specific transporter here will have to use some energy to allow the contaminant to go inside the cells. Usually, the plant doesn't want to make to absorb the contaminant, but it uses these specific channels for other essential elements, and some of the contaminants can act as the essential elements, and that's why you can have contaminations of the cell. So if we go back to general scheme of the uptake by the roots, if we look at the inorganic contaminants, such as the metals, globally, you will have a major flux which will be through the water uptake of the plant, and the elements will follow the flow. You can have also active importation of cations, so elements which are needed by the plant, such as potassium, magnesium, and so on. When they are uptaken, it is exchanged by proton, by H+. And so this will influence the composition of the soil solution. If we look at the plant root, the soil solution, the soil solid phase, the soil solution is really the place where the elements will be available for the plant root. So it's a central point of the system. And there are exchanges. And the plant, by its activity, modifies the composition of the soil solution. And you can have acidification of the soil solution, which will increase or decrease some of the processes of exchanges between the solid phase and the liquid phase. Again, if the root is taking up AN ions, the balance is that it produces HCO3 minus, which will also influence the pH of the solution. And lastly, the root is able to produce some organic acids, which have a big influence on the mobility of some of the trace elements. Some species are really specialized in foraging the soil, and they are able to mobilize essential elements for them, such as the iron. When it is missing in the soil solution, they are able to produce these organic acids that will facilitate the uptake or the dissolution of the iron or other elements like iron in the rhizosphere. So this is the main processes that are linked to the inorganic contaminant. And when you look at the organic contaminants, it's mostly through the fluxes of water that the organic contaminants will enter the root. So there is no specific exchange with the protons or HCO3 minus. But as some of the organic contaminants are lipophilic, and they will interact strongly with the various membranes of the roots. And so it means it's very difficult for the organic contaminant to cross the Caspary strip and to go in the upper part of the plant. Usually, organic contaminants are blocked and are more concentrated in the root and in the epidermis compared to the other organs of the plants. If we now look at the leaves, so if you make a cut in the leaf, you will have different layers, and at the external part you have the epidermis, and the epidermis is covered by a kind of wax membrane. So it protects the leaves against the external conditions. So it protects against high temperature, it protects against pollutants and so on. But it means that it's a kind of impermeable layer across the leaf. It's not impermeable, it's just less permeable. And so if we look at the leaves, in the lower part, you will find here some stomas, which are openings in the leaf. And these openings can be wide or more close according to the climatic conditions. So the plant is able to adapt the opening. But it means that there are openings, and so the air, there can be exchanges of air between the leaf and the atmosphere. And so if you have pollutants in the air, they can be absorbed directly through the stomates by the stomates and be absorbed by the neighboring cells of the stomate and go into the phloem. The phloem is alimented by the uptake of the plants and the activity of the chloroplast and so on. And so it's fed by these cells, and the phloem is the tubes that are able to transport the pollutant from the leaves to the other parts of the plant. So this is a main entry point for the pollutants that are in the air. But you know that sometimes you make fertilization by foliar application. It means that the particles that, or the compounds that you put on the cuticle are able to go through the surface of this cuticle. It's a slow process, but still a process that can occur, and that occurs. And so you can have wet deposit or a dry deposit of particles on the leaves, and they are able to go through the membranes at slow rate, but still it is for some plants important contribution to the contamination of the plant. So there are two main entries, one for the roots, one by the leaves, and between you have internal transfers that are occurring. So you have the xylem and the phloem fluxes that are occurring, and so there are redistributions of pollutants that are possible, but there are a lot of barriers to cross for the pollutants. So this is a representation of the cationic exchange capacity that are on the membrane. So these tubes are figuring the porosity. So the water here, this is here the inside of the root. You have macropores, micropores in a network, and the water and the elements are able to enter by these pores. So the bigger contaminants cannot enter, but the smaller can enter. And if they are charged, they might be retained by the charge, negative charge of the membranes. So this is for the internal pore. Here, this is the representation of the membrane. So you have phospholipidic membrane. It means that you will probably retain some lipophilic contaminant here and hydrophobic there. And also here it's a representation of the fact that the vacuole is an organ in which the contaminant can be stored by some specific cell. So some plants are specifically adapted to control and accumulate the toxic elements inside these plants. So to make a summary of the plant uptake here, you will have the roots and the leaves. The main entry is by water uptake, which is sucked and goes to the leaves. And in the leaves, your main entry will be gas uptake, wet deposition, or dry deposition, and contaminants can go down with the phloem. Also, there is a process that can be often important is when you have rain and the splash. So it means that the kinetic energy of the drop of water are disrupting the soils, and the particle are splashed on the plant, and it's kind of deposition of these plants. And so if the soil is contaminated, of course, it's a new way of entry to the plant. To sum up, for inorganic species, it is really important entry is the soil solution uptake, but there are also possibly dust deposition that can be important. So you have, in the ideal world, to clean the as much as possible the vegetables when you are growing them on contaminated sites. There is also transports of routes to aerial organs, but there are a lot of barriers and local storage, and it is very complicated to predict from one plant to another. The family of plants, the species, the varieties, cultivars are all factors that can make this process vary. And regarding the organic species, in order to be uptaken by the roots, they must be in solution. The lipophilic retention is very strong through the barriers. And so the leaf uptake is an important way of entry for this pollutant. And one process that is important too is that the pollutants are characterized, organic pollutants are characterized by the volatilization property. And so with the natural property of volatilization, they can volatilize from the soils to the atmosphere of the leaves, or also it can be enhanced by the activity of microorganisms. So if microorganisms are active, they can boost this volatilization process, and it can increase the risk of contaminations of the plant. So it means that when you want to characterize the soil and the transfer from the soil to the plant, you have the concept of bioavailability. So consider that the pollutant is available if he can enter to the plant. So bioavailability has been defined in various contexts. So it is more general than the plant. It's availability for living organism. When you are specific to the plant, you will speak about phytoavailability. And the idea is that a big part of the component of the pollutant in the soil is more or less strongly bound to the soil. And if it is strongly bound, it will be not available. Only a part of the contaminant will be available. This graph is probably mainly more focused on the metals on inorganic species rather than organic, but yeah, a lot of a big part of the elements in the soils, when you look at heavy metals, for example, is not available. And then after you have different pools in the soils, which are more or less strongly absorbing the elements, and so the more strongly the element is absorbed, the less you will find in the solution. We are speaking of environmental bioavailability or availability, sorry, environmental availability, when you are looking at the way or the relationship between the solid phase and the quantity that you will find in the pore water. Because the pore water can be taken by the plant, but it also can be leached to the aquifers, and it will not impact organic living organism such as the plant. But it can move in the soils and impact all the contaminant or the compartment. So we speak about environmental availability, and we speak about environmental bioavailability for the part of the contaminant that is able to go through the cell membrane. And then after, we speak about toxicological bioavailability for the part that pass through the membrane that is also able of toxic effect of on the plant. Regarding the factors that are affecting the bioavailability, the first compartment to take into account is the plant. Then we will look, there are the pollutant and the soil. Regarding the plant, of course, the plant characteristics and its physiological state. So is it functioning well, or is it does it a disease or not, and so on. Because the most important flux is water flux, and it is driven by the transpiration rate. So the more biomass, maybe the more area, surface area of the plant, the more it is able to transpirate, and the more the plant will suck water from the soil. So the transpiration rate is important. And then you have the, of course, the characteristics of the plant, so the phenotype characteristics, the size of the organs, the nature of the membranes that the pollutant have to cross, the porosity, so the size of the apoplasm, the cell characteristics, the root exudates, and so on. So there's each plant are different than the other one. So you have big family that are recognized by the legislation according to what is eaten, for example. But yeah, if you look at the plants, they have a lot of different properties that all are making a kind of variation in the bioavailability of the pollutant. Also, the plant can have tolerance or detoxification mechanism that are different from one plant to the other. And also, of course, the organ that you consider. If you are speaking about carrots, it's mostly the root that you are interested in, and it's easier to have pollution in these roots. But if you look at the tomatoes, it's a fruit, and it means that to go into the fruit, the pollutant uh, has had to cross a lot of barriers. So it's more difficult to contaminate the fruits than the roots in a general way. There are always a specific behavior. So that's first factor which affects the bioavailability. It's the plant characteristics. The second one is the pollutant characteristics. So it can be mineral pollutant, organic pollutant, dissociating pollutant or non-dissociating, charge element, non-charge, big molecules, small molecules, and so on. So all the pollutants have their own characteristics. I think you already seen that in other presentation. And then you will have the soil characteristics, such as the total reserve in pollutant, the reactivity of the constituent, it's kind of chemical properties of the soils, also the physical properties of the soil, such as the organization or the structure, which will influence the behavior of the plant and the accessibility of the contaminant. And lastly, the biological activity of the soil, which will have an important role not only for the fate of organic pollutant, but also for some metallic pollutant too. So that was the main part regarding the concept. I don't know if I make a first stop before going to the first part, the second part.
Yeah, thank you, Gilles. It was very interesting. I'm looking around. I did not receive any questions in the chat, but are there questions or remarks on this first part relating to the concepts of uptake to plants? I think your figures made it very clear, Gilles. So, okay, I don't
Took me some
see.
time.
That's okay. It worked well.
Okay.
Okay, so no questions at this moment, Gilles. So I propose you go on with the models.
Okay. So regarding the models, I think that's something you already saw today or yesterday too. It's that there are different kinds of model. Here it's just a reminder that when you are looking at a model, usually there are two big families. One is deterministic model and the other one is stochastic model. So it means that you have a kind of a random variation in the stochastic model. And in the deterministic model, you find also differentiation according to the degree of complexity of the model, with the empiric model, which are more like a statistical relationship between or correlation between two parameters, and mechanistic, which are going into the mechanism, the detailed mechanism of the processes. And of course, when you are wanting to make models, you have to make choice of the kind of model that you will use, because in terms of availability of data, it changes things a lot. As an example, we were working on the contamination of vegetables through a project 10 years ago, which was called the Polysol 2 project. And in this project, we sampled vegetables from gardens, private gardens, and we made this kind of simplest model, which is looking at the soil content and the vegetable content. Here, it's an example for the leaf vegetable. So we measure the soil content in some contaminants and also the vegetable content. For some of the methods that we followed, we saw that whatever the content in the soil, the content in the vegetable was always below the limit of quantification. There are others in which it was the inverse for the thallium and the molybdenum. You found them in the vegetable, but never over the quantification limit in the soil. So with this, you cannot do a correlation between the elements, and you just say, okay, the mean content of vegetable for molybdenum is that one, and that's all you can predict. You just take the means of the value that you measured. But when you have data which have been quantified either in the vegetable content and in the soil content, you can try to have a look at a prediction model. So what we had is, did we find significant correlation between the two? Yes or no? For elements such as barium, cobalt, copper, molybdenum, thallium, and fluorantane, organic pollutant, in the leaf vegetable, we did not find correlations, and so we could not make a prediction model. And for cadmium, nickel, lead, zinc, arsenic, and chromium, there we found some, and we could make this kind of model, which is rather simple. You have a model which predicts the trace element in the salad according to the trace element in the soil. So it's basic empirical model. One element or one parameter which is often found in order to predict the content in the plant from the content in the soil is called the bioconcentration factor, BCF. So it's something that you often encounter in the literature. So it's the relationship between the cadmium in the plant and the cadmium in the soils, and it is supposed to be constant. So here you have a relationship, and if you take the slope of the regression line, you have a constant. And this is an example in the literature which give you BCF values for some metals. If we look at the cadmium, we have 1.77 for the radish, 1.73 for amaranth, pumpkin less than 1, and so on. So you will find different kind of BCF in the literature. When you go more and more, you read more and more articles, and you try to see, is there consistency in the BCF? You will see that there are interval of variations of this BCF, and they can be very, very wide. This is a relationship that we made in our data, and we measured the cadmium in salads here, and here it's a prediction that was made with a model. We used a general model here rather than using a model of BCF. We looked at a model which is integrating not only the cadmium in the soils, but other characteristics such as the pH, the organic matter, the clay content, and so on. And so that was the first model that we had. In an ideal world, the points should be along this line. So it's this is observed and this is a predicted value. And here we say that there is some bias in the data, and we made another model more adapted to our data set. So the first one was a model that we found for another data set. We applied it, and here we just made the same analysis, but on our data set. And on our data set, we can fit it better than the other one. But the question is, with this kind of model, can you make generic model that you can apply everywhere, or a specific model? So this also shows that the bioconcentration factor is not in our data set, is not a constant, the bioconcentration factor. So the ratio between the cadmium in the plant and the cadmium in the soils depends on the other factor. So it's not, even for a data set, it should not be viewed as a constant value. So this is an illustration of an empirical model. There are rather few mechanistic model of the uptake of contaminant by the plants, because it means that first you need to make a plant growing model. So you need to be able to model how the plant is growing. It means that you need a climate model. You need also a soil water model to feed the model. And in the best case, you need a soil fertility module. Then after, you are going to have a look at the specific root behavior of the contaminants in the root. And so maybe you will need a kind of geochemical model in order to predict the composition of the soil solution. And then you need to be able to model the sorption of the contaminants through the various barriers inside the root zone and also inside the other membrane. You need to have a diffusion model of the contaminants through the leaves. You need to have a gas exchange model from the atmosphere to the leaf. You need to have transportation model between the different parts, and also maybe transformation model. So the plant might be able to transform the organic pollutants, and also microbiological model. So it means if you really want to have mechanistic modeling of this, you need a lot of equation to and processes to feed with data. And usually you are in the best case, you can go to hybrid solution between fully mechanistic and empirical model. I took the example because in Belgium, and it's the case in all parts of Belgium, in the 3 regions. They use S-risk as a model in the evaluation of the risk linked to the contaminated soils. And S-risk as a model of transfer of a module of transfer of the contaminants to the plants. So I took it as a model here. It can take into account some of the fluxes that I already showed previously. Usually, processes that can be taken into account is root uptake from the soil, leaf uptake of volatile compounds, solid deposit on the leaves, diffusion inside the leaf, translocation between the roots and the aerial parts. More seldom is the volatilization of the pollutants from the leaves. Because there can be also pollutants can go outside of the plant. They can volatilize through this tomato too. You can have also dilution of the pollutant according to the growth of the vegetation. And so the organ is growing, and the contaminant might be diluting by the growing of the organ. And also there might be biodegradation of the pollutant inside the plant, and usually this is not taken into account in the model. Also, in the model S-risk, there are two compartments. One is the soil, and the other one is the aerial part of the plant, where you will specifically target the organ that interests you. So it means that if you are interested in the tomato, you will the aerial organ will be the fruit of the plant. So if we look at the root uptake in the S-risk, see that the idea is that the model uses bioconcentration factor. So it means that you have to know the bioconcentration factor specific to your plant and your soil and your pollutant. So you can directly put your bioconcentration factor, and it will calculate the concentration in the roots according to the concentration in the soil. So the general equation here is that the content in the roots originates only from the soil. And so it depends on the concentration of the pollutant in the soil solution and the BCF that you have to specify. So this is an equation. Another one is when you don't have the BCF, he will calculate for the plant another factor, which will take into account the transpiration rate of the plant, another parameter, which is the partition coefficient between the tissue and the root, another coefficient, which is the loss and the elimination of the pollutant by the plant, and another one, which is the dimensions of the root. So it means that you have something which is a bit more complicated and which requires some data. So there is in the document that explains how the model works, you have some tables which fix the parameter values. So it means that when you use the model, it has been calibrated for very general use, and maybe they are not completely specific to your actual problem. So you will have some parameters. So some parameters that allows you to feed this kind of equation. Another point to take into consideration in the root uptake of organic pollutant is the importance of some of the parameters, which are the key OW, key OC, and the key H, which are characteristics of the pollutant. So it's a partition between the the solvent and the water when you have your parameter. And the key OC is the partition of the pollutant with the organic matter. It means that this parameter, which gives you the affinity of the pollutant to the organic matter. In the soil, you have organic matter. And if the affinity is high, it means that the pollutant should be fixed on either the organic matter or on the membranes. And another important properties of the organic pollutant is the KH, the Henry constant, which is a property of volatilization. The higher it is, the higher the pollutant is able to volatilize and go away with the atmosphere. So it means that you have a list of all these parameters that are given by tables and that feed the model. So maybe it looks like a more mechanistic model, but you have a lot of small buttons to make a switch in one direction or another one. If we look at the inorganic species, it's the same model as the organic contaminant. It's really BCF which will drive the content in the roots. And so we have a table, we have a tool that was made in a project which is called Sunisol. And in that tool, there's a list of BCF that are given for the various vegetable that can be found. So the relationship are really specific between the pollutant, the plant, and the soil. And so there exists some table that you can use, or you can either you find them in the literature, or you adjust your own data set in order to calculate this BCF. But again, it means that you take one BCF, which will make that you consider only one relationship between soil and the plant for your situation. BCF is kind of constant. Once you have fixed the soil properties, the plant, you have only one BCF that is calculated. After that, another factor parameter in the model of uptake is the above ground uptake, so through the leaves. And you have similar situations for organic or for inorganic. It's the same principle, that the concentration that you will find in the above ground part is a part comes from a translocation from the root. So this is VS here, VSG here. One is the plant concentration, which is linked to the deposition, wet and dry deposition on the leaves, and one contribution from the splash. And for the inorganic, are only considered the translocation and the splash particle. No, not the splash, so the VP. So it's the wet and dry deposition. And again, it means that you must be able to evaluate the particle deposition. And here you have an equation. We want to have a detailed look at it, but you can see that it needs a lot of parameters that you have to estimate. And if you cannot measure them, you should be happy that people already made estimation of these parameters before. So it means that here for the different plant, for each plant, you have a lot of characteristics, a lot of parameters to be fixed. So this is an example, one of the table in the model with the various plant characteristics. So it was mainly I told about metals and or inorganics and organics. There might be new contaminants, emerging contaminants that you are not taken into account by the models, but you should say question about the way it works. But usually it's the same principles. They to enter in the plant, they have to cross the same barriers, the same entrance, and you will have a mechanism for entrance through the root by the apoplasm, symplasm, and the entrance through the leaves by the stomatas or by going through the cuticles. So for the principle, it's exactly the same. The only things that can change is the parameters that you will find in the table. There's even nanoplastics can enter in the plant. Okay. So maybe stop in order to see if there are questions before going to the case study.
Yeah, okay. Thank you, Gilles. Are there any questions? We saw a lot of equations, a lot of data sets. It's indeed complex. There is no question on the chat, but I wait a few seconds to see if somebody raises hands or has a question. No, or it was very well explained, or it was too complicated. No, no. I'll leave it up in the middle. No, okay. I think maybe Nathan has a question. Nathan?
Sure, that's interesting. It's interesting that you mentioned the microplastics or nanoplastics. What contaminants would they behave like? Like I don't think they're cations like heavy metals, but where do they sit in the plant uptake world?
That's a good question. Indeed, I don't know exactly. I only studied microplastics in some master works here with students, and know that there are a lot of different kinds of plastics, and I guess they should behave like organic pollutants, but it depends on if they are charged or not. Do they have a surface charge and could be absorbed like the cations or not? I don't really know. I'm not a specialist in that, but
yeah. I guess they're hydrocarbons, right? They're just small, but they're bigger than atoms or compounds, but smaller than. Yeah, interesting. I guess.
Yeah, it's really the nanoplastics that can enter. But are they retained? How much? I don't know exactly. I don't know if they behave like lipids or not.
Yeah,
I really don't know.
I suspect there's a lot of ongoing research nowadays. I seem to see it in the news a lot. So probably busy area. Funding.
Yeah, the thing I can add to that is indeed there is a lot of research going on, because also in a lot of cultivation, some plastics are put on top of the soil, and they're also looking at how they degrade. I think one of the aspects they are considering, but then that's more the larger pieces of plastics, may also change some physical soil structures, so having porosity effect on that. If you go to the nano and especially the other plastics, what you also must consider is very rapidly they form some kind of a biocorona around them with all kind of bacteria and other stuff as well on it. So they change very rapidly as well. So also that could maybe enhance transport because they are not like more virgin anymore on the surface. But indeed, it's a big field of new science, and I think we will definitely in the coming years have more on this topic. That's to come, definitely. Yeah.
Yeah, I think we're all in the right line of work for just continuously emerging new contaminants.
Yeah.
Yeah, at the beginning, I was a bit skeptical. I said, "Oh, why should we worry about microplastic and so on?" But yeah, now the research showed that there are really transfers. They are able to go in a lot of bodies, and even said that you eat the equivalent of one bank card. In your body due to the contaminations by nanoplastic. So it's scary.
by the end of when? What was that stat? By the end of next week, did you say, or year?
No, I don't know on many time, but yeah, I think that we incorporate the equivalent of one plastic card of the bank card, but I don't know if it's on the lifetime or it's on the shorter. I don't know exactly, but even if it's on the lifetime, I'm not sure I want to have a bank card in my head.
Okay, I don't see any other questions. So indeed, Gilles, please proceed with your case study.
Okay, thanks. Yeah, the case study, it's about metals in the vegetables. And so, as I already told, we were part of a project which is called Polysol 2 at the 2010 approximately. And the idea was that we were focusing on the gardens in the areas which were affected by atmospheric pollution. So we're in urban environment. And if you look at this beautiful garden, you don't suspect that there might be problems of contaminations. But if you take that one, you will be, oh, I'm not sure I would take some salads from that garden close to. But yeah, these two situations are typical of what we find in cities. And it doesn't signify that this one is more polluted than the other one. So we were asking a few questions such as what is the level of soil contaminations in kitchen gardens? What are the levels of plant contaminations and what are the risks for human health? And so we at some specific area 8 sampling zone close to along the Meuse and the Sambre, and also one in the south of Belgium. So all these were known for large-scale atmospheric deposition of pollutant. And we sampled almost 400 gardens, and we took more than 1,000 vegetables in these gardens. And we made the analysis of 19 inorganic contaminants, almost 40 organic contaminants, and we also measured 9 soil characteristics. All the analyses were made by professional commercial laboratories and not from the university. So we had 4 types of vegetable: leaf vegetable, fruit vegetable, tubercules, and root vegetables. And at the first results, you just look at the soil properties in these area, what we see is that as it is a kitchen garden, the levels of organic matter are rather high. The clay content is medium, close to 15%, and the pH is pH KCl is close to 7, so it's rather high. So in this case, we can suspect that the metal contaminants should not be too much bioavailable, as the pH is a factor of precipitation of some of the metals. So at this pH, they should be less available than if the salts were acid. And organic matter is also a factor that can fix some contaminants. Yeah, so environment is keen to weak heavy metal mobility. And if we look at the trace element content, so the analysis, as the legislation is demanding, works on the total or pseudo-total content. So it is with the aqua-regia. And if we look at the columns are the various zones, so the various points. And if we look at the metals, we see that there are signature. In red, we have areas that are richer in cadmium, lead, and zinc. And they are really linked to close to Liège, linked to a former mining industry of processing calamine, and so it's a zinc ore and lead ore. They were processing that, and the cadmium was a component of these ores. And a very specific contamination, so higher level in arsenic in the south of the region. So after that, we had a look at the vegetables. But when we look at what the mean content in the vegetables, we see that we find the same signature. So the more in the places where you have more cadmium in the soil, you also have more cadmium in the plant. The same for the lead and the zinc, with sometimes some variation, such as in this area, it was rather low in lead in the source, but you find a rather high content in the plant. So we have the similar local signature with an effect of soil content. And so if you have an effect of soil content, you want to make a model of that. And the first model is the BCF. And so if we look at the soil cadmium here and the plant cadmium here, we plot the data for the lettuce. You see that if we had the unique BCF, we should have one single linear relationship between the points. Maybe it's not so easy to see. And we calculated the histogram of the BCF. So for every couple of soil and plant. And if you look at the histogram, you see that most are less than 1, but you can have some variation between some are less than 0.5. Most are less than 1, but you can have situations where you have BCF until more than 10. So you have a big variation in these in this BCF. In fact, I draw here 3 lines which are corresponding to different kind of BCF. So if we had to take one single BCF, none of these can represent here the behavior of the plant and the soil. So that's a limitation of the BCF. If you take the mean BCF for all the plants, maybe it's 1, but it means that there are some situations that you cannot well represent in the data. So if you make some now transformation, because BCF is on the data which are not transformed, if you make a log transformation, it begins to look like a linear relationship between the log transformed. And the equation is now that you have the log 10 of the cadmium in the plant is A plus B multiplied by the log 10 of the cadmium in the soil. So it also means that the log 10 of the BCF is dependent on the log 10 of the plant and the log 10 of the soil. And so that if you make the transformation, that the log 10 of the BCF is a function of the concentration in the soil. And so you should not consider BCF as a constant, but you can try to continue to work with the BCF, but predict him according to your data, not use middle BCF that comes from the literature. So we, after that, tried to see if we could have a better equation than only the prediction of the plant according to the prediction to the content in the soil, and we used the Polysol2 regression equation that was made on this data set. And we had a kind of relationship with a mean error of minus 0.4. It means that on our data set, we sorry, I forgot to say, we here worked on a specific data set that was not used for the calibration. So we took a validation data set that was independent from the Polysol2 calibration data set. And so it means that you have a mean error of minus 0.4, so there's a slight underestimation of the content in the plant with the model that was calibrated. Because you have a lot of data here surrounded here that are not well predicted by the model. It is not able to reproduce these very high content in the plant. So it's kind of buffering. It's typical, yeah. And so that was the first step. We had a model and with its accuracy. Then some years later, in another project in which we were focusing on group gardens, so community gardens and not kitchen. We made some analysis, and when we gave back the information to the people who were owning the gardens. Some were a bit worried in the Bressou garden because we found very high levels of soil contamination. And then there was a call for making another analysis, and we had a specific investigation on that garden. And so on the same garden, we had almost 60 plots, and we took 160 soil and vegetable samples in these plots. So we had a lot of data. And we just at that time, the only way to see if there was some compliance with the regulation, it was with the former regulation, and we could make some recommendation because we had some vegetables that were good to compliant to the regulations, and the other one which you should discard. And so the study continued with that gardens, and the idea was trying to make models of risk analysis. And so in the risk analysis models, you have to define the targets, the source of the pollutant, the targets, and the way of transfers. And so that was done for this garden. And at the end, we could have a series of recommendations to the garden and say, okay, here maybe more studies are needed. And the studies continued to occur, and it allowed to construct a web tool dedicated to the people who own kitchen gardens. And the web tool gives them information about the risk of transfer from the soil to the plant. So they can put values about the soil content, and they will have estimations of the plant content. And this estimation of the plant content, so there is a specific tool on Excel that was developed too, allows to calculate the part of the metals for arsenic, lead, and cadmium that they are susceptible to ingest due to the frequentation of the garden, due to the alimentation. So do they eat the plants for their gardens or not, and so on. And this model, of course, is based on equations that were built. And so we first begin with the equation from the Polysol 2 model. And during this project, which is still in progress, we continue to measure and measure and measure couples of plants for soil and vegetables. And so now the models have been adapted to more situations, and they change a little bit the equations. Then more recently, there were a change in the regulation, in the EU regulations regarding the levels of lead and cadmium that were allowed in the legislation in the food. And there were some spotlight that was put on the situation of market gardeners around Liège. And they found that they had some problems to fulfill the compliance of the regulation due to the change of regulations. And indeed, if you look at the mean soil cadmium, here it's a map of the mean soil cadmium that you can find in the municipalities. It's mean by municipality, and according to the existing data, of course. But you can have a look, and Liège is here, and there are some indications that the province of Liège has a lot of sites that have more cadmium than the rest of the region. And so there's a lot of market gardeners that are producing vegetables in that area, and they want to feed the city of Liège, which has a big initiative of which is called Feed Liège, Nourrir Liège. And so it makes them a lot of trouble as they are not allowed to sell their product. And what we had a look, we made some analysis in some of the gardens here, A, B, C, D. So that's a different gardens where we took samples, and this is the cadmium in lettuce, and in black this is the old regulation, and in red it's the new regulation, and we see that before, some of the gardeners were okay with the regulation, but some were not. But now, with the new regulation, almost every gardener has a problem. And so we were trying to see, can we find solutions for them in order to reduce the transfers of contaminants from the soil to the plant. And what we knew from the literature is that the pH might be an important factor. And it is confirmed by pot experiments that we had. Here it's the cadmium that you find in the salad. And it's decreasing when you increase the pH of soil. So it's not really new, it's well known. But the idea was to be able to quantify and to make experiments in the field in order to see if this is confirmed by the experiment. And so we had a lot of data now with the content in the soil and content in the plant. And we were trying to see if it follows the models of prediction. If we can model the relationship, we can, at the end, try to make some simulations in order to see what solution could help us to reach the target of the regulation. And so we made some prediction, and we used the Polysol 2 model to our data. You see that it really doesn't fit our data. And we have here a mean error of -4. The previous model on the calibration data on the validation data set was -0.4. Here we are 10 times higher as a mean error. So, yeah, the question was, can we have other models? We have the luck to have a wide data set, wider than Polysol 2. And we try to use it and model it and then predict it to our data set, and it was even worse. Then we tried other techniques, only multivariate regression model, but also more sophisticated, such as a random forest. And here, okay, it does not seem to perform very well. And so the question is still which other model we should use. And then we used a model from Sanisol, which was an evolution from the Polysol 2 model, which seems to work a little bit better. So we have a mean error of -2.4. And so the question now is it acceptable, and can we be satisfied with this kind of model, or should we go further? And of course, at the end, is it applicable to have this kind of bad estimation? At the end, we have a lot of variation on the estimation. Can we be satisfied with that? Knowing that what we have to predict is content in the plant which is rather low. So this is here the plotted of the differences between predicted and observed value with the last model, and see that globally we overestimate the content in in the low low content, and we underestimate in the high content. And so that's a question that we have now. Should we go further, or when do we stop when we make modeling? So maybe to conclude here, models of plant uptake are based on the use of BCF. The BCF should not be seen as a constant, but depends really on the context and the soil content. Building generic predictive models seems hardly achievable, while a specific model require a lot of data acquisition. If you want to make models to be free of not measuring, but if you need to put a lot of effort in making specific models, maybe doesn't worth it. There is still room for research in the field of plant uptake prediction, and meanwhile, the knowledge of uncertainty associated to the prediction is a prerequisite for decision making. We must know how confident we are in the estimation from the models, and does that fit with the regulation before taking decision on what can be done and what cannot be done. Thank you.
Okay, thank you, Gilles, for your nice overview of all the things we must consider related to plant uptake. And also, I thank you for sticking to the time. And that's also appreciated. Are there any questions in the audience on the plant uptake in general? I think this was more this session to give you an overview, because we will not consider it in the training, but it was more as a general aspect of things that you can consider, or how in the models the contamination and the plant uptake is considered. So we found it essential for you to know what the roots are, how this is calculated. And of course, if there are still questions later on, as we also said yesterday, you can always ask or send your email to Anna, and she will make it over to us.
I think, Giles,
yeah, Nathan?
maybe you can comment on this, but I think one of the important things to understand about these models is that they go into the development of the standards, the numerical standards generally. So if we're going out there and sampling an agricultural soil for mercury, and we're comparing that to a threshold value, like where does that threshold value come from? And these sorts of operations are what develop them, I think, is kind of the big picture.
Yeah, thank you, Nathan, for pointing this out. Okay, I don't see any more questions pumping up. So here I would like to thank you once again, Gilles, for your very nice presentation. And then I think we will see each other in more or less 1 hour, a little bit less than 1 hour, for then the last session of today, also related to modeling, but then more in soil and groundwater. So if everything is fine, I will see all of you within, let's say, 50 minutes, more or less. Okay. Thank you all.
Thanks.
Thank you, Gilles.